So, what is enzymology? Enzymology is the study of enzyme, their kinetics, structure, function, as well as their relation to each other. Enzyme occurs in all living systems and very necessary to support life. Enzymes are found in plant, animal, microbes, but in industrial point of view, microbial sources are the preferred. Amino acid sequence are important that are found in the active site of enzyme. Active site is an enzyme where the substrate molecule is bind. Active site found on the surface of enzyme. Basically, enzymes are chemical composition of enzyme. Enzyme basically are proteinaceous in nature except ribozyme, which is RNA in nature. On the basis of chemical composition, enzymes are classified into two parts, two different types. First is simple enzyme, second is conjugate enzyme. Simple enzyme composed of simple amino acid sequences, but conjugate enzymes contain protein part that is called apoenzyme and non-protein part that is called coenzyme. When non-protein part are organic in nature, then it is called coenzyme. When non-protein part is inorganic in nature, then it is called cofactor. When apoenzyme binds to its non-protein part, then it is form a complete enzyme, active enzyme called holoenzyme. Apoenzyme is the characteristics of enzyme. Enzymes are complex macromolecule with high molecular weight. They can accelerate the rate of biochemical reaction but do not start the reaction. Enzymes affect the rate of biochemical reaction but not the direction of reaction. Most enzymes have high turnover number. Enzymes are very specific for their substrate. For example, invertase is highly specific for sucrose only. It do not work on other sugars or lipid or protein. Enzymatic reaction is of course affected by different physical parameters like pH, temperature, because change in pH and temperature causes change in the protein structure, and protein structure is unstable or denatured. Denatured enzymes are not functionally active enzyme and cannot able to convert substrate into product. Regulatory enzymes are also again very important, which regulate the metabolic pathway. The example is allosteric enzymes. Allosteric enzymes contain two sites. One is active site where the substrate molecule is bind, another is regulatory site where the regulatory molecule or modulator molecule is bind. Modulator molecule may be positive modulator, may be negative modulator. Positive modulator enhance the affinity of enzyme towards its substrate. Negative modulator reduce the affinity of enzyme towards its substrate. Multi-enzyme complex. Multi-enzyme complex are the enzyme, single enzyme containing different active sites on their different subunits where the different substrate molecule is bind and different product is released. So single protein catalyze hold the multi-step enzymatic reaction. Isoenzymes are the enzymes which catalyze the same reaction but their amino acid sequence is different. They are encoded by different genes, they have different molecular weight but catalyze the same reaction. For example, lactate dehydrogenase enzyme found in 5 different isoenzymes. Enzyme kinetics. Enzyme kinetics is the study of the chemical reaction that are catalyzed by enzymes. In enzyme kinetics, the reaction rate is measured and the effects of varying the conditions of the reaction are investigated. The popular equation for enzyme kinetics is Michaelis-Menten kinetics, in which V0 equal to Vmax S upon Km plus S, where V0 is the initial velocity of the enzyme catalyzed reaction, Vmax is the maximal velocity of enzyme catalyzed reaction, Km is the Michaelis-Menten constant, and S is the concentration of substrate in molar. In this plot, this is the popular plot of Michaelis-Menten kinetics called hyperbolic curve, in which reaction velocity of enzyme catalyzed reaction is studied with the concentration of substrate. As the concentration of substrate increase, reaction velocity is also increase. In first order reaction, when the reaction all the active sites of enzymes are occupied by the substrate, then enzyme is saturated with the substrate, and we further increase the substrate concentration, there is no increase in the reaction velocity and enzyme reach its maximal activity, maximal velocity called Vmax. Half of the Vmax is called Km. So Km is the substrate concentration when V0 is equal to Vmax by. Why enzymes are important? Enzymes are important because of the cost effective production of end product for many industries like textile industries, food industries, detergents, biofuel production, and many more. Many enzymes are useful in therapeutic purpose. Enzyme inhibitor are also act as in drug. The inhibitor block a certain type of reaction and inhibit the growth of pathogenic microorganisms. Basically, inhibitor are three types: competitive inhibitor, non-competitive inhibitor, or uncompetitive inhibitor. Uncompetitive inhibition, their kinetics are totally different. Enzymes are also used as analytical uses. Enzyme used in biosensor for the detection of different type of analytes such as environment pollutants. Enzymes are also important for creating lactose-free dairy products. Immobilized enzyme is again very important. Immobilized enzyme are the enzyme where single enzyme is bind to a inert insoluble material to reuse the enzyme or to stabilize the enzyme. In an industrial point of view, immobilized enzymes are very important. Enzyme engineering is a novel field in which we change the structure of enzyme by changing one or two amino acid of the enzyme. This can also be done by site directed mutagenesis, where change are occur at the level of